Genome editing, also known as gene editing, is a cutting-edge technology that enables scientists to alter an organism's DNA. This process involves adding, removing, or modifying genetic material at specific points in the genome. One of the most revolutionary genome editing tools is CRISPR-Cas9, short for Clustered Regularly Interspaced Short Palindromic Repeats, and CRISPR-associated protein 9. CRISPR-Cas9 has gained immense popularity due to its speed, affordability, precision, and efficiency compared to older genome editing techniques. The CRISPR-Cas9 system is inspired by a natural defense mechanism found in bacteria. When bacteria face viral infections, they store fragments of the virus's DNA in their own genome, forming CRISPR arrays. This storage acts like a genetic memory bank, helping the bacteria recognize and respond to the same virus if it attacks again. When reinfected, the bacteria produce RNA segments from the CRISPR arrays. These RNA pieces guide enzymes like Cas9 to the invading the virus's DNA, enabling the enzyme to cut and disable the virus. If you're fascinated by the science of genetics, consider exploring The Gene, An Intimate History by Siddhartha Mukherjee. This book provides a deep dive into the history and future of genetic research. Find the link in the description below. Scientists have adapted this bacterial immune response for gene editing in laboratories. They design a custom RNA sequence, called guide RNA, that matches a specific target DNA sequence in a cell. This guide RNA is paired with the Cas9 enzyme. When introduced into cells, the guide RNA directs Cas9 to the precise DNA location, where the enzyme makes a cut. After the DNA is cut, the cell's natural repair mechanisms take over, allowing scientists to add, delete, or replace specific genetic sequences. CRISPR-Cas9 holds enormous potential for treating and preventing human diseases. Researchers are exploring its use in conditions caused by single-gene mutations, such as cystic fibrosis, hemophilia, and sickle cell anemia. The technology could also help combat complex diseases like cancer, heart disease, mental disorders, and even HIV. Looking for hands-on learning? Check out STEM and genetics kits for learning on Amazon. They're perfect for curious minds eager to experiment with real-world genetic science. Find the link in the description below. However, gene editing raises significant ethical concerns, particularly when it comes to editing human embryos or germline cells, egg and sperm cells. Changes in these cells can be passed down to future generations, sparking debates about altering traits like intelligence or physical abilities. Due to these concerns, germline editing is currently illegal in the United States and many other countries. Despite the challenges, genome editing continues to be a groundbreaking field with the potential to reshape medicine, agriculture, and beyond. As research progresses, the scientific community remains focused on balancing the promise of curing diseases with the ethical implications of altering life at its most fundamental level. Subscribe for more videos like this. See you in next video.